When I was studying biochemistry at the University of Dhaka, this was in the mid-1970s, the discipline of molecular biology was just at its infancy. But it had already generated the excitement that is still there today. It is a captivating field which seeks to understand at the molecular level all the fundamental issues that govern anything living, from the smallest of microbes to the largest of the mammals. I was naturally attracted to it. And this intoxicating research on genes and their products that I carried out during my PhD, and armed with the tools of the trade, I was ready to undertake my future work on jute molecular biology. Choosing jute as the field of my research was to a great extent to honor my father, who had already passed away by then. I knew about his love for the country, because of which he had wanted his second son. He had sent his second son to study Jute. This was in the late 1950s to the Institute of Jute Technology, an institute of the Kolkata University. My father believed that this crop, which had such a profound influence in the economy of a country, had to be studied inside out. I wonder which other father at that point in time would have wanted his son to study jute, and that too in a different country, and not want him to become a doctor or an engineer. For us, Bangladeshis, jute is a symbol of our identity. It is not only a crop which produces fibers and fetches a lot of foreign currency, it is our national icon linked to the adage Sonar Bangla. In the mythical Bengal, around which most of our folklores are constructed, we had these undulating rice and jute fields swaying in the wind, and also fields of jute fibers drying out in the sun, golden in color. It is a carbon neutral crop. It gives back much more to the soil than it takes in as a fertilizer. Simply put, jute is a wonder crop. And we are very lucky that Bangladesh is home to the best quality jute grown in the world. And one best thing, the good news that we have, as of 2023, Bangladesh has regained its position as the number one producer of quality jute. This is despite the decline in production. In order to make room for growing more food crops, jute is being pushed into inhospitable terrains where it has to cope with a lot of adverse conditions like waterlogging, aridity, salinity. Therefore, in order to have a handle on the crop so that we can make improvements in it, it was necessary for us to understand jute at molecular level, to understand the molecular landscape of jute. When we started work on jute, we did not have any idea how to isolate quality DNA from the jute leaves. We didn't even know how to analyze those DNA. But what we had, and in fact we were quite lucky to have, is a big germplasm collection. This was made by the International Jute Organization. This is a collection of jute seeds and seeds of allied fiber collected from all over the world. This is the gene bank that we have at the Bangladesh Jute Research Institute. There was this one characteristic among this germplasm that caught my attention. There were some seeds in this collection which could germinate at 16 degrees centigrade. We know jute loves growing in hot, humid weather, and therefore most jute will not germinate below 20 degrees centigrade. But these seeds were germinating below 16 degrees. Apart from the traditional uses of jute that we have, which requires retting is a process of submerging the whole jute plant and allowing it to rot under submerged conditions so that the fibers come out of the stalk. We have to think of other alternate value-added uses of jute. Even the water bodies are shrinking, and we know we do not get the rainfall when we are supposed to. Therefore, farmers often have to resort to using shallow, dirty water for the retting purposes. This degrades the quality of jute and does not fetch as much revenue. But we could use the green jute as a source of paper pulp, which would not require any retting. But for this, we need to grow jute round the year so that we can feed the paper industry. That is the reason why I was interested in those seeds which could germinate below 16 degrees centigrade. Jute seeds which can germinate at 16 degrees centigrade and those which we cannot. But the markers that we used for jute were so non-specific that we were very skeptical about its success. But these same markers worked wonderfully in our hands. It landed us on a jute DNA, jute gene, in fact, which we think is in some way directly or indirectly linked to low temperature tolerance. For us, it was like hitting the jackpot. Beginner's luck, you may call. Luck to some extent, but what we have in aplenty is the bright young minds, our students, who often make up for the limited resources that we have. This initial success of isolating a jute gene ignited the passion in my students, the passion that is required to think big, to think out of the box, and to embrace uncharted path. The first isolation of one jute DNA gradually led us to find more genes which were having different functions. And one gene at a time, we were getting the confidence of handling big data. So the dress rehearsal was over, the stage was set, and we were ready to do the mega project, the sequencing of the jute genome. Therefore, when late Dr. Maqsoodul Alam joined hands and the government came forward with generous funding, we were ready with quality jute DNA obtained from plants of single seed descent to ensure purity. And also we had the skilled manpower which could undertake this daunting task. Announcement of the draft jute genome created quite a stir. In fact, it was announced by the Honorable Prime Minister Sheikh Haseena in the Parliament. It made the headlines. So, what did the jute genome do for us? Gave us the confidence to handle big mega projects. It gave us the manpower, and in many cases, it also provided the necessary infrastructure. Following the jute genome, we have the genome sequence of the many national icons, the Hilsa fish, the jackfruit, the black Bengal goat, to name just a few. While we were doing the analysis of the jute genome sequencing, trying to find out genes which would be useful in genetic improvement of jute or introducing one trait into the other, we were aware of this roadblock. This roadblock was that the two jute cultivars that we have, which the farmers use, one is Corchorus oletorius, which is tosha jute, and the other Corchorus capsularis, which is white jute. These two jute are cross-incompatible, and also they are recalcitrant to dedifferentiation and in vitro regeneration. That means jute is unresponsive to tissue culture. In this backdrop, in my lab, we developed a technique for transformation, which is tissue culture independent. It mimics a condition that occurs naturally, a condition in which agrobacterium, a soil bacterium, which infects plant. Agrobacterium infects plant at the wound site, and it introduces a part of its own DNA into the plant. Now, in our hands, a transformation protocol that works quite well. And we use this transformation protocol to reduce the lignin content in jute. When we had the jute genome sequence at hand, found the pathway through which jute makes lignin. Lignin is a component of the plant cell wall, which allows the plant to stand upright, which protects the plant from pathogens. But the jute fiber has so much lignin, and the fiber becomes so coarse because of the lignin that we cannot consider as a textile fiber. So it was important uh, to think about reducing the lignin content so that in future we can consider it as a textile fiber. So this transformation protocol, in which we tried to reduce the lignin content of the jute, worked quite well. This is now we have 7 generations after a transformation that the transgenic lines are still showing reduced lignin content. At this point, let me shift gears and tell you about my unexpected encounter with jute endophytes. An endophyte or an endosymbiont is a bacteria or a fungus which lives in a plant. Most of their life, they live in the plant without causing any damage to the plant. They are considered as paying guests. And in return for the food and lodging that the plant offers this fungus or bacteria, they provide a lot of benefits to the plant. They provide biomolecules which help the plant in growing better. It helps produces bioactive compounds which help the plant defend itself from pathogen attack. It helps the plant in many other ways. So my intro to the jute endophytes was made when we were analyzing the jute DNA sequences, and we were getting a lot of sequences which matched bacteria. My first reaction was, this must be a contamination made by the person handling the DNA. But later on, I realized those in fact were DNA of the bacteria or the fungus that live inside in the jute plant, and they were being extracted through the process that we were using for extracting jute DNA. We now know plant is not a standalone entity. In fact, no eukaryotic organism is a standalone entity. We, even human beings, we are meta-organisms because we house trillions of bacteria and fungus within our body. So the plants also have many bacteria and fungus living with them, and that is what we were getting when we were isolating the DNA. And we all are extended phenotypes of the microorganisms that live in our body. Our preliminary analysis of those endophytes proved them to be promising agents of many bioactive compounds. So we resorted to isolating many of these microorganisms from the different tissues of jute, leaves, then the whole plant, the bark, the roots, and we were getting a number of bacteria and fungus. We now have a good collection. There are many known endophytes in their collection, and there are many in our collection which are unknown, which has not been previously reported as endophyte. And those endophytes on a characteristic, we were surprised to find the activities that have. One fungus that we isolated was producing Taxol. Taxol is a multi-billion dollar cancer drug. And there was another bacteria which was producing lantibiotic. It is a type of antibiotic in which there are some amino acids which are not non-protein amino acids. And they were active against many human pathogens which are multi-drug resistant. Then we had a bacteria which could prevent the plant from a phytopathogen, which is Macrophomina physiolina. This Macrophomina physiolina is a plant pathogen which infects more than 500 plant species. So this bacteria was making a special kind of compound called pyrrolonitrile, which was helping the plant in preventing itself from being infested by the phytopathogen. The many challenges that the human being that we are facing at the moment, an increasing population to feed, an altering climate, an increased dependence on pesticides, and an emergence of microorganisms which are multi-drug resistant. And answers to most of the issues we can find, answers to those we can find through jute and the endophytes. I'm confident that for many more years to come, jute will remain to be our source of pride, our source of happiness, and our source of prosperity. Thank you.